Good morning to all. Today we're going to see about part 2 of pharmacodynamics. In part 1, we had discussed about what is the mechanism of drug action, what are the targets of drug, and different enzymatic activity, what is receptor, what is ligands, of the basic definition of all this we had seen. Now in part 2, we're going to see exclusively about receptor pharmacology, which means the receptor is a major component in maximum of a drug is being active. Okay. So we'll move on. First, it is what is receptors? Okay. So as you all know, receptor is a macromolecule or a binding site located on the surface of inside the effector cell that serves to recognize the single molecule or the drug and initiate the response to it. But itself has no other action, which means the receptor per se doesn't have any action on it, but it used to accept the ligand to bind on it and produce an effective drug. That is the primary thing which is happening. And most of the receptors or proteins normally serves as receptor for endogenous regulatory ligands and the physiological receptors. So as we discuss the receptors part, we should also know about what is ligand is, because the ligand is a molecule or a substance or a drug which goes and binds on top of the receptor, which makes the effect of a drug. So any molecule which attaches selectively to a particular receptor or sites, okay, that indicates affinity or ability to bind without regard to the functional claim. It just has to go and bind with the receptor. So that molecule is called as a ligand. Receptor is a molecule which is present on the top of the cell or inside the cell or whatever. It is all protein. The ligand is a molecule which goes and binds only to the receptor without any functional change within the receptor. Okay. Clear now what is a receptor and what is a ligand. Okay. Then the thing is you have to know about what are the transducer mechanisms of the receptors existing here. Okay. So here when you go, it is we have a different type of receptors, which is a G protein coupled receptor, ion channel receptors, transmembrane enzyme linked receptors, and transmembrane JAK-STAT binding receptors and receptors in the regulating molecules. So as you know, in the G protein coupled receptors, so the first thing is we wanted to see about the G protein coupled receptors. Okay. G protein coupled receptor, as you see in the picture, it is a very coiled structure. It looks like very complicated, which is a 7 helical membrane structure, which is binded around the insides, and it has a 2 binding sites. One is the agonist binding site, and the other one will be the G protein binding site. Okay. So in this GPCR, it has a 3 components. One is the alpha, another one is beta, another one is a gamma. So 3 subunits we have, and always alpha subunits, it's bind to the G protein, which is GDP. According to the which G protein it is goes and binds this alpha subunits, we are distinguished as GS, GI, GO, and GQ. Okay. GS means it is stimulates. It stimulates some enzyme or enhance some activity. Okay. I means GI means it is inhibitory. It goes and inhibits some enzyme on an ongoing process of the reactions. Will get inhibited. GO and GQ. Okay. Clear with the structure of GPCR receptors. And so according to the GS, what it goes and stimulates, it stimulates adenylyl cyclase enzyme and calcium channel opening. Okay, GI when it comes up, it is inhibit adenylyl cyclase enzyme and potassium channel opening. GO it is calcium channel inhibition, which is serotonin 5-HT1. And GQ it is phospholipase activation. These are the components where all acts on the G protein receptors. Okay. So GPCR per se, as I said, in that it has a 3 subunits. It is alpha, beta, and gamma. Alpha subunits is always bind to some G proteins, which is a GDP. Whenever this alpha subunits is bind to GDP, it tags as inactive state of the receptor. This receptor is not yet activate, so it is inactive. When this GDP it converts into GTP, then only this G protein coupled receptor it's will activate, and then this alpha subunit and this GTP will dissociate and from beta and gamma subunits. So we have the 2 components now. One is the alpha subunit with GTP and beta and gamma subunits. So whatever it may be the G protein link, which is GS, GI, GO, GQ, this will be the common for everything. When alpha subunits with GDP, it is inactive. When it converts this GDP into GTP, then it will become active. After getting the receptor is activated, then alpha and GDP will be dissociated separately from beta and gamma. So these two components will have their own specific action within the cell. Okay, this will be the basic knowledge you have to know about the GPCR before going into the specific type of GPCR receptors. Okay. Then what are the effector pathway? As I said, in case of GS, GI, GO, GQ, a specific enzyme and also some channels will be linked together to get the desired effect of the receptor activation. Okay. First, we will be seeing about adenyl cyclase, cyclic AMP pathway. So, in which kind of GPCR receptor, as we say, adenyl cyclase and cyclic AMP pathway? GS or GA? It is GS. Yes, GS stimulation. Okay. As you see here, this is the receptor, which is a beta receptor, which is meant on the left side, which is a beta receptor. It has a agonist binding site. The pink color, which is marked as agonist binding site. So adrenaline will go and bind on that agonist binding site. After this alpha and GDP, it is binding, which is the receptor is inactive. And GDP, when it's converted to GTP, it will become active kind. As I said, it will get dissociate this alpha and GTP go. Here it is, we are talking about GS. So this component will go and activate adenyl cyclase enzyme. And what it happens with adenyl cyclase enzyme is it converts ATP into cyclic AMP. Okay, this adenyl cyclase enzyme which converts ATP into cyclic AMP, this converted cyclic AMP which goes and activates phosphokinase A. What will happen when the phosphokinase A gets activated means two things will be there. One is like allows the calcium to get inside the cell. Another one is allow the calcium into the cytoplasmic reticulum. The two things. When we discussed about the GS transducer mechanism, what we had seen with the GS, adenyl cyclase enzyme activation and calcium channel open. When you see in the left side corner, a calcium channel is there. From there, the calcium is getting inside into the cell. Yes, due to the increased, due to the activation of phosphokinase A, it causes more increased calcium within the cell. Okay, so because of that, the troponin is get activated, a better excitation contraction complex, which causes increased cardiac contractility. So, one activity due to the activation of phosphokinase A, it is increased cardiac contractility activity. The other thing, what happens is it is an activation of phospholamban. What it means, what does the phospholamban over here? Here is the phospholamban goes into a faster sequestration of calcium in the sarcoplasmic reticulum. What happens? Whatever the calcium is getting inside the cell through calcium channel, those calciums will be stored inside the sarcoplasmic reticulum. So what happens? There is a downfall of calcium level within the cell, which causes relaxation. So that causes faster relaxation of the cell. Okay. This is how acts when the GS of G protein coupled receptor is get activated. So here is we will stimulate adenyl cyclase enzyme. Due to the stimulation, ATP it is converted into cyclic AMP. This cyclic AMP goes and activate phosphokinase A, and then this will go and activate troponin and phospholamban. Due to troponin and the activation of calcium channel, it causes faster contraction. Due to the activation of phospholamban, more sequestration of calcium into the cytoplasmic reticulum, which causes relaxation. Clear? I hope everyone clear with the GS subtype of GPCR receptor. And second thing we'll see about what happens when the GI subtype of a GPCR receptor. Okay. In the right side, you have a N2R, which means muscarinic 2 receptor. Okay. That is the GI kind of a receptor. So when acetylcholine, the red color on top of an N2 receptor marked as agonistic binding site, where acetylcholine goes and binds on that N2R receptor. What happens is this, as I said earlier, the alpha subunits with the GTP is inactive. When alpha with GTP gets activated and dissociates the beta and from the gamma. Correct? So here the dissociated beta gamma here will goes and activate a potassium channel. Okay, this alpha and GTP goes and inhibit adenyl cyclase. Okay, due to inhibition of adenyl cyclase, what it happens? There is no conversion of AT2 cyclic AMP, no activation of phosphokinase E, no activation of troponin and phospholipid. There is no contraction and relaxation process will be happening. Clear? So this is how it is acts on. And what happens? The dissociated beta gamma from the muscarinic receptor. Okay, this beta gamma it activates potassium channel. So potassium channel, what it does, it allows the potassium to move inside the cell or moves out of the cell. Yeah, someone can respond. Yeah, it's correct. It is allows the potassium to move out of the cell. So when the more potassium is going out of the cell, which causes hyperpolarization, which allows the resting state of the cell. Okay, this is what happens. So here we discussed in adenyl cyclase pathway GS and GI subunit of the Kawsalliya receptor. Anybody having any doubt, you can raise at any point of time. I can explain you further. Okay. Next, we'll move on to the other subtypes of the Kawsalliya receptor. Okay. Other will be phospholipase C and IPTAG, which is GQ. So here we have this green color one, H1R written as histamine receptor, which is GQ activity. So here yellow color meant agonistic binding site where histamine will go and bind on it and it will get activated. Alpha and GTP will be dissociated from gamma and beta. And what happens when alpha and GTP goes? It goes and activates phospholipase C. So what does this activated phospholipase C means? It goes and cleaves phosphoinositol biphosphate, okay, into DAG and IP3. You know that DAG and IP3 is a second messenger. It causes more number of reactions within the cell. So this IP3 goes and activates the calcium channel, which causes releases of more calcium, more entry of calcium within the cell, and more calcium will come. And this calcium will go and activate MLCK, this is myosin light chain kinase, and CCP calcium calcium protein kinase. This due to activation of all this, it causes more contraction of the muscle. Okay. And what happens? This is due to the IP3 activity. When DAG goes, it is phosphorylate more proteins. Okay. IP3 DAG goes and activates phosphokinase C. This phosphokinase C goes and phosphorylate proteins, more protein phosphorylation. This is how the GQ receptor is being active in this. Okay. So we discussed here is GS is activation of adenyl cyclase, enzyme, and also activation of calcium channel. Second thing is GI, which is inhibition of adenyl cyclase enzyme and potassium channel activation. And GQ, which is activation of phospholipase C and the activation of calcium channel. Okay, clear with mechanism whatever existing between the GPCR. Okay. Second, the third thing is we'll discuss about channel regulation. How all these channels being regulated within the GPCR. The activated G proteins, which is GS, GI, GO, whatever it is, it also can open or close or inhibit the ionic channels specific for calcium and potassium, okay, without the intervention of any second messenger. So GS, what happens? It opens the calcium channel in the myocardium and the skeletal muscles, okay. GI and GO open the potassium channels in the heart and the smooth muscles as well as inhibit calcium channels even. Okay, this is how the channels are regulated within the GPCR receptors. Apart from GPCR receptor, what other type of receptor existing in the body is ion channel receptors. What are those ion channel receptors? Those are ligand-gated ion channels, which is selective to the specific ions, which is allows ions, all these ions to move out of the cell or enter into the cell. It depends on their osmolarity on all this. So agonist directly operates the ion channels without the intervention of any complete protein or second messengers. Okay. And onset and offset of the response of the ion channel receptors are within the milliseconds will be there. So agonist which goes and binds and open the channel, what happens? It causes depolarization or hyperpolarization, which causes changing in the cytosolic ion composition, depending on the ion flows through the channels. Okay. Examples for the ion channel receptors are nicotinic receptors, GABA receptor, and glycine inhibitory amino acid receptors. Okay, these are all the ion channel receptors. And we'll discuss about the transmembrane enzyme linked receptors, what it means, what the name itself means, something which is transmembrane enzyme linked receptor, which is receptor where it is present normally outside the cell or inside the cell. So the receptor is present in the transmembrane, which has an enzyme activity. Okay. So this has, as in GPCR, we mentioned how many helical structures? The 7 helical structure with the 2 binding sites will be there. Here, you have a single helical structure and it is present transmembranely. Outside the cell, you have a hormone binding site, and inside the cell, we will have the intracellularly, you will have an enzymatic activity. Okay. So it is a large extracellular ligand binding domain connected to a single transmembrane helical peptide chain to an intracellular subunit having enzymatic property. What happens when the hormone, this enzyme, transmembrane enzyme receptors are very highly specific to the hormones like insulin, epidermal growth factor, nerve growth factor, all those factors. So this is very important. When the hormone gets bound to the extracellular ligand binding domain, then this receptor which gets activated, what happens is it gets dimerization of the receptor. Okay, it gets dimerized and also phosphorylation of the tyrosine residue. Through that way, are various effects of the receptor and also the drug effect also you will get. Clear? This is how a transmembrane enzyme linked receptors acts. The fourth thing is transmembrane JAK-STAT binding receptors. Okay. What it is JAK-STAT means? JAK means what? Janus kinase. So this receptor also is present in a transmembrane. It has a 2 component. One is a JAK, which is called as a Janus kinase, a STAT, which is called as signal transducer and activator of transcriptions. When the signal you get, there is a more activation of transcription. That is what it means. As like a transmembrane enzyme receptor, this is also a single helical structure. It has an extracellular ligand binding domain, and intracellular you have a JAK-STAT in place of enzymatic activity, which we seen earlier. Okay. This cytokine, the most specifically here is cytokine is the ligand which goes and binds over the receptor. This cytokine binds to the extracellular binding domain, which induces the receptor dimerization. Okay, the dimerization which activates intracellular domain to bind free-moving Janus kinase molecules. Okay, because this is more available within the cell, so it goes and binds the Janus kinase molecule into the receptor intracellularly. This activated JAK phosphorylate tyrosine residue, okay, on the receptor, which then bind the STAT, which is signal transducer and induce transcription. This tyrosine residue of the STAT also get phosphorylated by JAK, and phosphorylated STAT, it will get dimerized and dissociate from the receptor and go to the nucleus and to regulate the transcription. It targets it. This is how the transmembrane JAK-STAT binding receptor it works. Okay, so you have to know about this kind of factors. Fifth one will be the receptor regulating the gene expression and transcription factors and nuclear receptors. So here you know, everywhere we had seen about the transmembrane receptor, which is a receptor which is present in the extracellular or over the transmembrane. Here its name itself means a receptor which is present within the nucleus. So here we have to know more about what is the structure of the receptor. Okay. So as you see on the topmost in the image of the dropper side, you have a carboxy end and also amino end. You have a 2 region. One is the steroid binding domain, another one DNA binding domain. This 2 binding domains, steroid binding domain and DNA binding domain, which is connected by the hinge region. Okay, hinge region, and in the DNA binding domain, you have a 2 zinc fingers. Clear? When this receptor is binds to the HSP90, whenever the HSP90 is being bind to the receptor, it causes inhibitory action over the receptor. It doesn't go and do any other action by on itself. So when the steroids or whatever it is, ligand, it goes and binds over the receptor hinge region, it will causes the dissociation of HSP90. So no more inhibition will be there within the receptor. So this binding of steroid to the GR dissociates HSP90, removing their inhibitory elements on it. And then what happens is, then it goes and tries to the nucleus, which causes dimerization of the receptor occurs, which translocates into the nucleus and binds co-activator or co-repressor protein and interacts with the DNA sequence, which is called as glucocorticoid responsive elements. Okay, glucocorticoid responsive elements is very important to get for the transcription and translation processes. This is how the receptor acts when the nuclear receptors are present. So we discussed about Kawsalliya, ion channel receptor, and transmembrane enzyme receptor, transmembrane JAK-STAT receptor, and receptor regulating the gene expression, which is the nuclear receptors. This is example for the nuclear receptor. You can take thyroid receptors and also steroid receptors. Okay. So here you have to know about all these things, and the take-home message for the receptor it is, as we discussed, affinity and intrinsic activity, whatever it may be a property, it is agonistic, antagonistic, inverse agonistic, and partial agonistic, you will have affinity within everything. And with the intrinsic activity, agonist you have a maximum intrinsic activity of 1, and inverse agonist you have a 0 to -1, partial agonist you have a sub-maximum, antagonist you have a nil intrinsic activity. Then you have to know about what is drug synergism and antagonism. So when two or more drugs are given simultaneously or in quick succession, they might be indifferent to each other or exhibit synergism or antagonism. Okay. So there's all the synergism antagonism way it applies means when you combine two drugs together. It does not, you can't discuss this topic when you're giving a drug alone. Okay. When you combine two drugs, how they are sharing their property each other, how they are helping it out each other, or one it's pulling it down each other, like that only. What the human does is in a similar way the drug also acts. Synergism means it is having two types of synergism. One is the additive, another one is a potentiation. Okay. Additive means effect of a two drug. When you combine the two drug together, the effect will be the same direction and simply adds up. Okay. Effect of drug A and plus B is equal to effect of drug A plus drug B. Example, when you combine aspirin with paracetamol as an analgesic and antipyretic, the effect will be the same when you combine or when you give separately or two drugs together. Okay, that is called synergism. Second will be a supra-additive or potentiation, which means the effective combination is greater than the individual effect of the components. Okay, so effect of drug A plus B, the effect will be more as compared to the drug A, effect of drug A and effect of drug B. Okay, example, a levodopa and carbidopa. Where we are giving levodopa and carbidopa? Yes, we are giving for the Parkinsonism. What happens in the Parkinsonism? What is the pathology behind a Parkinsonism disease? There is a depletion of dopamine in the brain. Okay. So we have a dopamine depletion, which causes we have to supplement dopamine for them. When we give levodopa, definitely it will release more dopamine within the body. But into the brain, it is releasing when it comes, it is a question mark. Okay. It causes peripheral decarboxylation. So very less amount of the dopamine only reaches to the brain, which is not enough to treat or to ameliorate the symptoms of Parkinsonism. Okay. That is why we combine the levodopa with the carbidopa. The carbidopa, what it does is it inhibits this peripheral decarboxylation of levodopa. Okay. That is why the efficacy of combining the levodopa and carbidopa for the Parkinsonism disease is more as compared to when you give a levodopa alone or carbidopa alone to a patient. Okay. And third thing we have to see about antagonism. So we had seen about synergism, which is when you combine the drug two drugs together, they have the same line of action. Antagonism is opposite. When one drug decreases or abolishes the activity or the action of another, they are said to be antagonistic. Okay. Effect of drug A plus drug B, it is less than the effect of drug A plus effect of drug B. So when you come talking about antagonism, you may have a two types, which is physical antagonism. Based on the physical property of the drug, they will go and inhibit or abolish the action of another component. Okay. For example, when it takes as a charcoal, which absorbs alkaloids and prevents their absorption. Whenever you go through with the poisoning, we wanted to stop them to absorb into the body. So we are giving the charcoal, it will go and bind the poisoning component, whatever it may be, and prevent the absorption of the drug or whatever it is. Okay. So it is used highly for the poisoning. This antagonistic property we are using it for the medical treatment. Okay. Second thing is chemical antagonism. Chemical antagonism means when two drugs react chemically and form an inactive product. Okay, which is KMNO4, which oxidizes alkaloids used for gastric lavage. Okay, this is also like for the poisoning and all this type. What is physiological? We had seen about physical antagonism, chemical antagonism. Here it is a physiological antagonism, which means the adrenaline, bronchodilation, bronchoconstriction, vasodilation, constrictions, it's all happening in the physiologically within our body. The drugs, how much it is happening. Adrenaline causes bronchodilation due to the activation of the beta-2 receptor. And histamine causes bronchoconstriction, action on the H1 receptor. Glucagon and insulin, which act on the blood sugar. Okay, so these all are the examples of physiological antagonism. Clear? So what we see here is synergism. In synergism, we have additive and supraadditive potentiation, and in antagonism, we have physical antagonism, chemical antagonism, and physiological antagonism. These are all the dose response of receptors. Okay. This is the receptor antagonism. So what we have is antagonism is, you know about a competitive antagonism and non-competitive antagonism. What is competitive and non-competitive? As a receptor structure as mentioned here in the resting state, there is a receptor. You have a two binding site within the receptor. One is the receptor binding site. Okay. Another is the allosteric site. Okay. Whenever the receptor goes and binds produce an effect, the receptor binding site it will come. Whenever the agonist also goes and binds on the receptor binding site. Okay. When the competitive antagonism happens is, with some molecules or antagonists, it goes and competes for the receptor binding site and produce no effect, no response. Okay. That is a competitive antagonism. You have two types with the competitive antagonism, which is the equilibrium type and non-equilibrium type. Example for the equilibrium type, which is pentolamine. Non-equilibrium type, we have a phenoxybenzone. Okay. So this is how activity it all happens. And in a non-competitive antagonism. So competitive antagonism means you have a equilibrium type and non-equilibrium type. In a non-competitive antagonism means you have a allosteric antagonism, flattening of DRC, and diazepa equilibrium. These are the characteristics and what are the features which are existing for the non-competitive antagonism. So what we completely know about here with the receptor is, what is receptor means? It is all the receptors are proteins. And next thing is receptor. It's whenever the ligand molecule which is goes and binds on top of the receptor and produces effect. What are the types of the receptor we have? We have a GPCR receptor. Under GPCR, we have GS, GI, GO, and GQ. Then ion channel receptor, transmembrane enzyme linked receptor, transmembrane JAK-STAT, and nuclear receptor. So I have clearly explained to you how each individually all these receptors are being acted and being produced. Clear? And then we were discussed about a dose response of the receptors. One thing is they have a synergistic activity or antagonistic activity. Under synergistic activity, we have a what? Additive and supraadditive or potentiation property. Okay. In antagonism, we have physical antagonism, chemical antagonism, physiological antagonism. And one more thing is competitive antagonism and non-competitive antagonism. This is all you people has to know about various type of receptor. This is a very basic of your pharmacology. When you know about the general pharmacology, that you are receptor chapter where in forthcoming classes, autonomic nervous system or maybe a systemic pharmacology or cardiovascular respiratory, this all will help you out. You have to know how the drugs are being acted. These are because majority of the drugs are acting through a receptor or the enzymes. When you're going on for the receptor level, you definitely have to know about all these factors to be aware of. It makes you people easy to understand a lot of concepts and knowledge within them. Okay? I hope everyone is understood well. If you have any doubt or anything, you can raise questions and comment. Always will be. You always read and chapter well and revise more frequently. That will help you people to recollect more exams and also for all competitive exams. Okay. Thank you all. Thank you so much. And read well and stay attentive to the class. Okay.